All right. Hi. So this is going to be the second half of the sugars biochemistry lesson that we had last week. We're going to talk. I'm going to focus on polysaccharides and then on sugar digestion. Right. We also had disaccharides. Just really quickly to list those off. The disaccharides are sucrose, maltose, and lactose. But polysaccharides have more to talk about, so we're going to dive into them. And so we have two types. We talked about glycosidic bonds, alpha and beta glycosidic bonds in the last video. So we have polysaccharide chains that are made of alpha glycosidic bonds, and we have polysaccharide chains that are made of beta glycosidic bonds. So as we remember from last time, we can break down alpha, we can't break down beta. So that's one major difference. Starch and glycogen are alpha polysaccharides. So we can break them down, and in the case of glycogen, we can also build it back up. Why do we break down starch instead of just leaving it if we can, use it? One of the reasons that we break it down when we eat it is so that it can cross into our body, because we can't absorb this huge starch molecule. Let's go ahead and go into what a polysaccharide might look like. So I've actually drawn these with beta glycosidic bonds. So I'm going to go ahead and ignore those for a second. So glycogen, I wanted to go into branching because it seemed like that was something that was a little bit confusing in class, and we kind of went over that for a while and didn't get a clear answer. So imagine each of these dashes that I'm making is a single glucose. And so after 8 to 10 glucoses, 4, 5, 6, 7, 8, then we will get a branching. So glycogen branches every 8 to 10. So then there will be a special one that allows it to branch. A special enzyme will come in and make a branched glucose. So this glucose is bonded to 2 other glucose. Then these go out, and etc. until it's 8. And then this will branch 2, okay? So after 8 to 10, it'll branch again. After another 8 to 10, it branches again. Now, this is on purpose in glycogen. The reason we want this in glycogen, if you remember, glycogen is how we store our glucose, and we want it available rapidly for muscle contraction and regulation of blood sugar if it's in the liver. So the liver will not be doing muscle contraction, obviously. The liver has glycogen, it'll put it into the bloodstream. And so it stores it as glycogen for ready access. Now, why does it branch? The reason that it branches, just for help in understanding, is because we have an enzyme that can clip off one glucose at a time, and we have a lot of them, so it could happen a bunch of times, but if you only have one end and there was no branching, then this whole glycogen chain could only have one enzyme at a time going, here's one glucose, here's one glucose, here's one glucose, and we wouldn't get glucose cut off as fast as we need it. But if we branch, then we can have, here's an enzyme over here that's acting on these ones. There's an enzyme for each terminal branch. Okay, so instead of having one at a time in this diagram, we have 1, 2, 3, 4, 5, 6, 7, 8 at a time. Magnify this by like 1,000, and you can see that you're getting like 8,000 glucose per short unit of time per few milliseconds, rather than 1 glucose per few milliseconds, because you have a lot of endings for those enzymes that can't fit in the middle. They can't like take this one. That one is in the middle of a chain. The enzyme can't do that. It can only do it from the end of the chain. And since you have so many ends of chains, you can pick off glucose from all of them, and you get much faster introduction of glucose into the system. So that is why glycogen is highly branched. I know that was an issue in class where it kind of went backwards and forwards. Glycogen is a highly branched, and more highly branched than cellulose, a highly branched polysaccharide. Okay, hopefully that's cool. So moving on, and we can break this down. So this is alpha, okay? And we store this, we make this via glycogenogenesis. We make this in the liver and muscle cells. That's where we store glucose and glycogen. Everywhere else, if glucose arrives, we pretty much break it down, or maybe turn it into like RNA or something, but we don't make glycogen, except for the muscles and the liver are the major ones. All right, so, really quick, cellulose, on the other hand, would be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, and then it would branch. So hopefully that makes sense on the difference of branching. And yes, cellulose can be a larger molecule, as she said in class, but with a given amount of glucose, glycogen is more highly branched. All right, so, beta polysaccharides. Cellulose is found in plants. It is not digestible because it has a beta glycosidic bond, which is what we have right here. It goes from an upwards facing hydroxyl on carbon 1, mark carbon 1 here, and it goes to the always downward facing hydroxyl on carbon 4 on another glucose. And this particular bond is what we're naming, and it is a beta glycosidic bond. And we can't break that. So we call this fiber. And this would go on and on and on, and you'd have another one here, and another one here, and another one here, and it would be cellulose. Chitin is exoskeleton of bugs. It has amino acids in it, and she mentioned it as an example, so I'm bringing it up here. It's something else that we would also call fiber. It's another form of beta glycosidic bond. It has obviously some different characteristics. It's used as an exoskeleton. So if you think about how the shell of a bug, that's chitin. And so that has a different texture and a different stiffness and so forth than cellulose. All right, so I think that's polysaccharides. Hopefully that's kind of a good overview. Now, moving on to the digestion, okay? So, I'll go ahead and do that there, I guess. Digestion. All right, so first, there's a nose. This is a mouth, okay? First, you eat, okay? You eat food. Inside your mouth, you will chew on the food. There are two things that happen in the mouth that are important for, we're now talking about digestion of sugars, taking them in, and there, now it's not creepy. We're taking them in and then we're getting them ready to be utilized. And what does that look like all the way down to glycolysis? That's what we're going over now. Two things happen in the mouth. So I would say that the first thing you want to remember is mouth. Okay, so the mouth has chewing or mastication, and then it also has salivary amylase. And we can tell by the last three letters on amylase, ACE, it's an enzyme, it breaks it down. This particular enzyme, called salivary amylase, has a pKa of 6.8. Okay, that means it functions best at a pH of 6.8. So that starts breaking down starch into, I believe it's maltose or mannose. But anyway, it starts breaking down sugars in your mouth. Now, when they get down to the stomach, that's my stomach, okay, duodenum there. When they get down to the stomach, the pH in the stomach is like 2 or 3. It's really low, and so that deactivates the salivary amylase. So the next part is stomach with a very low pH of like 2 or 3, okay? So after the stomach, so all the salivary amylase has been deactivated, which is why we have another amylase in the small intestine, because if it was still working, it's like, why the heck is there another amylase in the small intestine? Well, it's because the salivary amylase has been deactivated by the low pH. All the hydrogen, free hydrogen, which is what low pH is, we all know, all the low hydrogen has affected this and salivary amylase. So I'm going to try and make this next part bigger because I can tell it's not big enough right now. All right, small intestine. Okay, in the small intestine, we have, small intestine, SI, we have pancreatic amylase, which gets dumped into the small intestine. I believe it's the jejunum, but it's ileum, jejunum. It's the early part of the small intestine, which is the jejunum. And so the pancreas dumps in pancreatic enzymes, including pancreatic amylase. The reason I wrote pancreatic enzymes is because there's actually other enzymes that we'll later want to know. And if you just remember the pancreas dumps a whole lot of enzymes, I call them panzymes because why not? It's how I remember it. It includes pancreatic amylase, okay? So all the sugars that are down here, all the polysaccharides are being broken down by these enzymes to monosaccharides, single units of glucose or fructose or whatever they were. Most often we're talking about glucose. Those chains, here's a chain, okay? That chain gets diced up, and so then it's all single units. And those single units, we are able to use a transporter, which is called SGLT2, SGLT2, which transports monosaccharides into the blood. And it goes via, here's a cell. This whole thing is a cell, and it has a transporter on the other side. So the sugars go into this cell via sodium-dependent glucose transporter 2, SGLT2, and then there's another transporter on the other side which puts it out into the bloodstream. So you have glucose into the bloodstream, okay? So I'm going to erase this so that I can make bigger pictures, so hopefully, I know the lighting's bad and I know it's small, but hopefully it helps. I aim to get better with the lighting and all that jazz in due course of time. Alright, so let's move on. Alright, so it's in the blood. Here's the blood, and I'm going to make glucose blue dots. Alright, here's glucose, because I think you'll be able to see it better. Glucose is floating through the blood. Now, the first thing that really happens after you eat, your pancreas is going to notice, whoa, so let's just say that this right here, honestly I haven't seen the pancreas drawn a whole lot, so forgive me. This is the pancreas, and it sees that there's been an increase in blood glucose, okay? There's way more because we've just taken a whole bunch in through our small intestine, and it's been put into the blood. Now the pancreas is like, okay, my job is to help regulate this. And so it's going to put out insulin. There's something called a beta cell inside the pancreas, and that is what makes insulin. Insulin goes into the bloodstream, and I'm going to make it a star here. Insulin. Insulin is a hormone, a blood-borne hormone. And so it's going to go, hormones talk to things, that's what they do. So a hormone is going to go through the bloodstream, and everything that has a receptor specific for insulin is going to get the message from insulin. And it just so happens that insulin is one of the hormones that just about everything has a receptor for. Insulin can stick, it has a protein to stick to on just about every cell and organ. So it's going to stick to receptors on the outside of cells, just cells everywhere. I'll pull out green since we're going to go to cells. Anyway, and let's say that this is a cell. Here's its little receptor. And then insulin will bind to that. And now, once insulin has bound to that, this cell will put out a glucose transporter, probably GLUT4. GLUT4, okay? And that is to start taking glucose out of the blood and into the cell. This could be a muscle cell, this could be a liver cell, it could be a whole lot of things, okay? So that's lots of cells. Now, mentioning liver, there is something really important that the liver does. So we're going to have our bloodstream kind of weave around and go down here. All right. So this is still blood. Down here, let's make a liver. Okay, I promise it's a liver, okay? So, the liver is going to have, it's going to take up glucose as well, okay? So it'll have receptors and it'll have the binding and glucose will come in. Now, in the liver, yes, there's probably some of the same kind of thing that's going on over here, which is the beginning of glycolysis for these other cells. And the liver needs a little bit of that because, I mean, its cells have to stay alive. So they're going to do some glycolysis, just like all the other cells. And that's done using an enzyme. Once the glucose goes in, the first enzyme, and that is called hexokinase. Okay? And that's the one that starts the glycolysis thing. Glycolysis reaction, which then produces ATP and makes the cell able to use the energy that is stored inside glucose bonds. The liver, however, has a second enzyme that is only going to be super active if there's a freaking ton of glucose, because glucose doesn't stick to this enzyme very well, and so you have to have a lot of it to hit it, so that some of it will hit that enzyme in its specific site perfectly, and then it will grab it. Okay? More simply put, if you have a lot of glucose in the blood, then this enzyme turns on. Okay? And the enzyme is called Glucokinase. That is the one that is just in the liver, Glucokinase. I will go ahead and put GK because my liver is still too small. I need to get skilled at writing big, right? Ostentatious. All right. And I'll write up here HK for hexokinase. And you can have another, so these are our little categories here. This, I would just say pancreas and cells, just general, and then liver. This isn't quite how I organized it, but hopefully this can help you pause the video and see if you can remember, oh, what happened in the mouth? Oh, what happened in the stomach? What happened in the small intestine? What happened in the pancreas, the cells, and liver? However you choose to remember, it's great. All right, so glucokinase will take the glucose that's being pulled in via those glucose transporters that we've talked about, GLUT4, and it will pull it in. This is in fed state. It'll pull it in, and with glucokinase, it will do some glycolysis, and that in the liver happens, the glycolysis is happening not per se because the liver is like, oh, I need energy so bad. This time it's happening because we want to lower the blood sugar, because having high blood sugar for long periods of time is not good, and so the liver is helping to regulate the levels of blood sugar. All right? So now I'm going to move to what happens if we are no longer in fed state, okay? So the glucose is going to go down, and we're moving on to something. This is kind of the end of the lecture that she sort of went into on her actual sugars lecture. Now, I'm going to just, this was fed. Now we're going to talk about fasting or starved, okay? All right. So, oh, there's one more important thing with fed. One more important thing with fed is that while there's a ton of glucose, there's also glycogenogenesis, meaning we're making glycogen as well. And so there will be, I guess it doesn't come from glucokinase, but glycogen will be being formed in the liver and in the muscles. Which makes sense. I mean, you have a ton of glucose, you're trying to get rid of it, you want to store it somewhere rather than just trash it. And so you're going to make glycogen to lower the blood glucose. Now, in the fasted state, we have that glycogen built up. Okay, so let's expand our liver here. Okay, let's say that this is glycogen, because glycogen is, you know, it's got a chain thing going on every 8 to 10, and so, alright, so all this, all that right there, that's glycogen. Now, in the blood, if the blood glucose has gotten really low, and so that one dash right there, that's all the blood glucose that's out there. We're also going to have the pancreas still. Then the pancreas does one thing, and then the liver does one thing. Those are the two things you want to know about, okay? So the pancreas is going to put out something called, it's another hormone, and it's called glucagon, because the gluc is gone, which I believe Yakira said. So, fantastic. And, yes, so glucagon triggers the liver. So this goes and talks to the liver. This is the pancreas. This goes and talks to the liver and says, hey, there's no glucose out there. Fix this. So then the liver starts breaking down its extensive stores of glycogen. And we get more blood glucose, which means that the red blood cells in the brain can continue to use glucose, and it's all good, okay? So, we've gone over three things, really. Three major things. Polysaccharides, up there. Sugar digestion and fed state reactions to it, because when you're eating it, obviously you're in the fed state. And then 3, what happens when you move out of the fed state? The short story is, in fed state, you take it in, you store it in the liver and use it everywhere. And insulin's an important part of that. And then in the fasted or starved state, fasted state especially, because in starved state you've run out of glycogen. So I guess this is what moves you from the fasted state into the starved state, is when you run out of glycogen. That's like the trigger. Okay, well, we are now in the starved state because there's no more, no way to make more blood sugar, okay? So in the fasted state, the hormone is glucagon, which causes glycogen to be broken down in the liver and put into the blood. And this is important for the brain and red blood cells to get glucose to function well and properly. The brain can use other things, but it really prefers glucose. Okay, hopefully that helps. Cheers.